Alright, so this video is for my botany quiz over the movement of water and solutes in the plant body. So to start things off, first question, describe the major types of cell transport. So there's active and there's passive transport. Active transport requires ATP to move things in and out of the cell membrane. Um, and there's passive transport. Passive transport basically is an effortlessly, like, there's no ATP being expended at all, just stuff moving in and out of the cell, no problem, right? And then there's subcategories to both of those. For active transport, there's exo and endocytosis. And exo and endocytosis, basically both of them involve pieces of the plasma membrane, the cell membrane. So with endocytosis, that basically brings things into the cell, right? With endocytosis, it takes a piece off of the not piece off, but basically the cell membrane basically reaches up and pinches around some molecules and pulls them into the cell. And then exocytosis basically is kind of like the opposite of that. It has the membrane wrapped around molecules inside the cell and then shoots them out. And then that's endo and exocytosis. And then for passive transport, there's osmosis, diffusion, and facilitated diffusion. So with osmosis, that basically has to do all with water. Water, so basically it's water that moves in and out of the cell, right? And that one includes like moving things from a high to low concentration. It's concentration gradient. And yeah, so that's osmosis. Osmosis is just moving water from high concentration to low concentration. So like inside the cell could have a low concentration of water, and then there's a high concentration of water outside of the cell. So that means that with osmosis, more water is going to go into the cell effortlessly. So there's no energy being expended there. And then there's diffusion. Basically, diffusion is just a bunch of random random molecules, molecules moving at random inside and out of the cell, I think. Yeah. And then there's facilitated diffusion, which is basically, it's like diffusion, right? Random molecules going everywhere, right? Random movement of molecules. But there's a carrier protein. Yeah, there's a carrier protein that is basically inside the, not inside, but on the cell membrane. That basically is like a little hole that funnels all those little guys inside. It uses a carrier protein, but it does not expend any energy. It's basically like a big hole, essentially. So yeah, that's passive transport and its subcategories. And so that answered the first part of the first question. The second part is, how is understanding cell transport in plants important? So understanding cell transport in plants, that's really important because that's basically how plants get water and other minerals, I think. Yeah, but mostly water. We're gonna be looking at water. Or we looked at water. So that's why it's important, because it basically explains or basically shows or explains how water and other stuff goes inside and out of this plant cell. So that's the question one. So question two, what would happen to a plant in a hypertonic environment? I have a diagram. Something to help me out. So basically, in a hypertonic state, that's when there's not a lot of water going around, right? So a whole lot of solute, but not a lot of water. So basically, in plant cells specifically, when there's not enough water going on, or when water just exiting the cell and none's coming back in, the cell becomes plasmalized. And plasmalization is basically the inside part of the cell, the center of the cell, basically just shrivels up. It basically just shrivels up into itself, but it's still connected to the cell wall. And it leaves these stringy, weird little connections because it can't really separate from each other. And so that would decrease turgor pressure in the cell, basically affecting the external, which is right here. This sad little guy is a victim of a hypertonic environment. He is pretty much limp completely. There's no turgor pressure there holding him up at all. He cannot go straight. He cannot stand straight up because there's not enough water to basically put more meat or more stuff in between these spaces between the middle of the cell and the cell wall, basically to fill it out. So there's not enough turgor pressure there. There's not enough water to help it out. So that's what happens to a cell. That's what happens to a plant in specific. This happens to a plant. This is what happens to a plant on the cellular and external, um, on the external level. So that's my question for answer 2, or question 2. For question 3, I guess we can already see the big guy right here. Is the stomata. So explain the process that occurs to open and close stomata. So basically, to open, these guys use these dudes out here. These are guard cells. These are the ones that facilitate the opening and closing of a stomata. So basically, to open, water has to come into them and they'll swell up and they'll open up. It's pretty cool to see. So yeah, water goes in, they open. Water goes out, they close. So how that happens, there's 2 ways. There's active and passive transport. Here's passive transport, first of all, because it's the easiest to explain. Basically, water, there's a lot of water out here, right? Or not out here, but in here. There's a lot of water outside of the guard cells, right? But not enough inside these guys. Like they're still closed. So to open, through the process of osmosis, it just travels right through that cell membrane. Water just goes right back in. And to close, it goes right back out. But that feels pretty random, you know? Well, I don't know. I don't know. But there's active transport. With active transport, basically a little bit more complicated. You need the somata, basically the guard cells, right? Let's say there's a lot of solution out here, right? A lot of different business out here, and a lot of water in here. So what the guard cells would do is that they just forcibly take in some of that solute inside here, and then that just forces water out, right? It takes solute in and has water out, basically closing it. And then the opposite, it'll have solute in and then shoot it out. Well, not shoot it out, but it'll come out, and then water will come back in to open it. And this is what they think happens with plants when they're like from different parts of the day. During, like, it basically like helps control photosynthesis, or basically it's just it helps plants like basically time out or space out when they're like photosynthesizing or expelling or taking in gases. So that's stomata. And so that's the first part of that third question. And then the second part is, why would regulating stomata be important for a plant? Like I said, this would probably be really important for a plant throughout the day to basically control when it's not basically photosynthesizing, or when it's not photosynthesizing as hard. I don't know if that makes any sense, but that's what I got from the lecture for that. So yeah, that's all I got. That's all I got for that question, and that's the last question. So yeah, basically, yeah, this was my entry for the quiz for botany, and all of the information was from the lecture. I tried to put my own little spin on it, but yeah, thank you. Yeah, it's over.